Why is Black American DNA so unique? August 4, 2023, Cambridge, Massachusetts. A geneticist sits in front of a glowing monitor. On that screen, DNA sequences pulled from 27 sets of human remains, excavated from an iron forge in Maryland that ran on enslaved labor for over 7 decades, from 1774 to 1850. She runs a single search query against a database holding the genetic profiles of 9 million living people. She presses enter. 41,799 names come back. Let that land for a moment. 27 graves. Nearly 42,000 living relatives found in a single afternoon from a single search. You've probably heard that Black American DNA is unique, maybe more than once. You've seen the headlines, watched the reaction videos, heard it repeated until it stopped sounding like a claim and started sounding like fact. But here's what almost nobody tells you. The word unique in genetics is not a celebration. It is a measurement. And every measurement has a cause. What that cause actually is, and what it reveals about one of the most consequential events in recorded human history, is what this video is about. The Catoctin Furnace study didn't just find living descendants of 27 enslaved individuals. It found something embedded in the biology itself, something the historical record was designed to erase, but physically could not. The ancestry profiles of those 27 individuals were anomalous, even within an already extraordinary population. Their genomic signatures traced most strongly to the Wolof and Mandinka people of Senegambia, and to the Congo of what is now Angola and the Democratic Republic of Congo. That matters because the majority of enslaved people brought to the Chesapeake region came from Igbo and Yoruba populations in present-day Nigeria. These 27 individuals didn't match that majority pattern. They were outliers within a population already defined by outlier characteristics. 3 of those 27 individuals carried copies of the sickle cell gene variant. 4 carried a red blood cell enzyme deficiency called G6PD. Neither of these is a medical footnote. Both variants cluster specifically in the malaria-endemic coastal zones of West Africa. Their bodies had encoded, at a molecular level, exactly which part of that coastline they came from. No shipping manifest captured that information. No colonial ledger recorded it. The genome did. Silently, completely. This is why the Catoctin study matters beyond genealogy. 27 graves yielding 42,000 living matches isn't a historical curiosity. It is a forensic event. The genome isn't just a record of who these people were. It is the only uncorrupted account of what was done to them, and we've barely started reading it. Before we go further, I want to ask you something directly. Think about your own family history. Is there one branch, one origin, one gap in the story that you've always wondered about but never found a real answer to? Drop it in the comments, because by the end of this video, you're going to understand why that gap exists and what your DNA might actually be holding that no family story ever could. A research team at 23andMe does something that sounds straightforward but had never been executed at this scale. They pull up the Transatlantic Slave Trade Database, a historical archive of over 36,000 documented slaving voyages, and place it side by side with genomic data from 50,000 living people across 12 countries. The question is simple. Do the genetic patterns in living descendants match what the shipping records would predict? In several critical areas, they do not match at all. The study, led by researcher Micheletti, revealed that Nigerian ancestry, specifically Igbo and Yoruba genetic markers, is significantly overrepresented in Black Americans relative to what the historical shipping logs would suggest. At the same time, Senegambian ancestry is dramatically underrepresented. That gap is not a data error, it is a biological signal. Because more than 1.8 million enslaved people from the Senegambia region were documented in those shipping records. The ships recorded them leaving. The DNA didn't record them arriving, not proportionally. The working explanation among researchers is deeply uncomfortable. Senegambian-descended enslaved people appear to have been disproportionately placed on malaria-endemic plantations across the American South, where they died at elevated rates before having children. Their genetic lines ended not on the ocean, but in the soil they were forced to work. And when they died without descendants, their genetic legacy died alongside them. The uniqueness of Black American DNA is not just shaped by who was taken from Africa, it is shaped by who survived the destination. Standard historical narrative frames the slave trade primarily as an act of forced transportation. The genome frames it differently. It shows the slave trade was also, unintentionally but measurably, an act of biological selection. Not design selection, but selection enforced by the deliberate placement of human beings into lethal environments. The chromosome doesn't weigh intentions, it records outcomes. What makes this finding so striking is this: the historical record was designed to be incomplete. Names were erased, ethnic origins were stripped, languages were destroyed. That was the architecture of the system. But the genome cannot be confiscated. It doesn't care whether a plantation owner recorded the origin of the person he claimed to own. The DNA preserved that information in a language no legal system could seize. The shipping manifests tried to reduce people to cargo. The genome kept a different kind of ledger. And that ledger shows what the cargo manifests were too efficient to record, who didn't survive what came after. Charleston, South Carolina, 2022. Geneticists and archaeologists, working alongside the Gullah Society of Charleston, a community whose history is directly tied to this site, are processing DNA from 36 skeletal remains uncovered during construction near the city's historic port district. The individuals are African descended, late 18th century. They were buried a few hundred meters from what was, for most of the colonial period, the busiest slave trading port in North America. The team prepares 18 low-coverage genomes, and what those genomes show immediately is something no one had anticipated. Among all 36 individuals, buried in the same ground, at the same site, at roughly the same historical moment, the researchers cannot identify a single genetic family relationship. No siblings, no parent and child, no cousins. In any burial population of 36 people with natural community structure, you would expect to find relatives. There are none. These people did not come from the same families. They likely did not come from the same villages. They may not have shared a common language. They were, genetically, strangers to each other. That is not a random observation. That is forensic evidence of a policy. The deliberate fracturing of African family units, separating individuals from biological kin, mixing unrelated people from different ethnic and linguistic backgrounds, was not a logistical side effect of the trade. It was operational doctrine. Eliminate shared language. Eliminate community bonds. Eliminate the foundation from which organized resistance could grow, and it left a precise signature in the genome. When you build a population from dozens of unrelated individuals drawn from across dozens of different ethnic communities, you don't create a depleted gene pool. You create what geneticists call founder heterogeneity, a population assembled not from a single origin community, but from an extraordinarily wide cross-section of unrelated lineages. The result is a gene pool with baseline genetic diversity higher than almost any other population of comparable size in the Western Hemisphere. The uniqueness is real. It was engineered, not for the benefit of the people it was imposed on, but as a mechanism of control that produced an unintended genomic consequence. Shomlaka, Cameroon, approximately 8000 BCE. A rock shelter above a valley that will, thousands of years later, become the probable ancestral homeland of the Bantu-speaking populations, the people whose descendants will eventually be loaded onto ships. In this shelter, 2 children are buried together. Their genomes will not be sequenced until 2020. When they are, the results force a fundamental revision of where the story actually begins. Researchers published the Shomlaka findings in Nature. What the genomes revealed was a deep West African forager ancestry, distinct from modern Bantu-speaking populations, distinct from known Central African hunter-gatherer lineages, something older than both. A genetic substrate predating the agricultural revolution in Africa, predating the Bantu expansion, predating any civilization or political structure that appears in the historical record. One of the 4 Shomlaka individuals carried haplogroup A00 on his Y chromosome. Here is why that matters. A00 is the deepest branching human Y chromosome lineage currently identified by science. Its estimated divergence from all other human male lines is over 340,000 years. Anatomically modern Homo sapiens, as a defined species, emerged somewhere between 200,000 and 300,000 years ago. The most recent common paternal ancestor of the Shumlaka individual and every man alive today may have predated our species as it currently exists. This was discovered 5 years ago. It received almost no mainstream attention. The standard narrative of Black American genetic uniqueness begins in 1619. The Shumlaka data pushes the origin point back 340,000 years. And the genetic thread connecting that Cameroonian rock shelter to a living person today is not a metaphor. It is molecular. It is measurable. It has never been broken. The Middle Passage did not interrupt an ancient lineage, it transported one. The Jolof Empire, modern Senegal, 15th century. The Wolof people, organized into a functioning state with its own military, trade networks, and diplomatic channels with Portuguese merchants arriving on the coast, become one of the first West African populations systematically targeted by Atlantic slave traders. Within decades, Wolof men and women are being transported west in significant numbers. 5 centuries later, a genomic database finds Wolof mitochondrial signatures in the DNA of 27 Marylanders who died before the United States was a generation old. The Wolof didn't vanish from the record. They survived inside it, in someone else's continent, in someone else's language, written into someone else's official history while being excluded from it entirely. But the Catoctin finding opens something larger than one group's story. The Wolof are still present in West Africa today. The Mandinka are still present. Their communities, traditions, and languages survive. But when researchers map the genomic profile of those populations within West Africa, certain lineage frequencies appear lower than historical population estimates would predict. And in some cases, those specific frequencies appear in African American genomic databases at rates 3 to 4 times higher than in the modern West African source populations. The diaspora did not only originate from Africa. The diaspora has become a repository of African genomic history that Africa itself can no longer fully access. The research relationship is now inverting. African Americans are no longer only subjects of studies tracing African ancestry. Their genomes are becoming primary source material for reconstructing West African population histories that were disrupted by the very trade that created the diaspora. The laboratory has a new orientation. The data flows in both directions now. Here is the finding that the entire preceding evidence has been building toward. In 2020, the Micheletti study didn't only track ancestry, it tracked the ratio of African female to African male genomic contribution across 12 countries in the Americas. And in parts of Central America, that ratio reaches 13 to 1. In parts of South America, 17 to 1. 17 African female genomic contributions for every 1 African male contribution. No natural migration event produces a ratio like that. Not famine, not disease, not any demographic process that operates on a population without external control. Pre-existing West African genetic diversity, however extraordinary, does not explain 17 to 1. Because that ratio is not a feature of West African genetics, it is a feature of what happened to African men after the ships docked. The bottleneck is not in Africa, it is in the Americas, and it was created deliberately. The counterargument that Black American uniqueness simply reflects the pre-existing extraordinary diversity of West African populations actually strengthens the case when you apply this number. Because if that pre-existing diversity is real, then what was suppressed in the Americas was not ordinary variation. It was the male genetic contribution of one of the most genetically complex human populations on Earth. The scale of the loss is compounded by the scale of what was lost, and the consequences did not end with emancipation. A study published in PLOS Genetics in 2016 tracked the genomic footprint of the Great Migration. Approximately 6 million Black Americans relocating from the South to northern and western cities between 1910 and 1970. The sex-biased genetic patterns created during the 17th and 18th centuries, the specific haplogroup distribution asymmetries shaped by the slave era, were still visible in regional American genomic data as recently as 1970. The biological imprint did not end when the legal structure did. The Micheletti study's language is measured and scientific. It describes patterns consistent with a history in which interracial mating primarily occurred between mothers with African ancestry and fathers with European ancestry. The translation requires no scientific training. The 17-to-1 ratio is not a statistic about women. It is a statistic about men. It is a genomic measurement, precise, reproducible, cross-referenced across 50,000 individuals in 12 countries, of how completely the reproductive future of African men was suppressed. Not by mortality alone, by a system architecturally designed to prevent their biological legacy from continuing. European male lineages replaced African male lineages in the gene pool, not through natural population dynamics, through a structure of power that controlled who could form families, who could raise children, and whose genetic line would survive into the next generation. 50,000 people, 12 countries, the same pattern everywhere. African women contributing to the gene pool at dramatically elevated rates, despite men comprising over 60% of the enslaved population transported across the Atlantic. The names of those women were not recorded. Their ethnic origins were not recorded. Their children's names were not recorded. But the mitochondria kept the accounting. Across 3 centuries, across a dozen nations, in every population studied, the female mitochondrial signal is strong. The corresponding paternal Y chromosome signal from the same source populations is a fraction of what it should be. Every Black American man watching this carries a Y chromosome that passed through that filter. That is not heritage in the conventional sense. That is the record of selection, and the conditions of that selection were not natural, random, or neutral. We are now at the edge of what the science has confirmed. I will tell you exactly where that edge is. Among the genomic patterns researchers have flagged in studies of African American ancestry, there is a category of variants appearing at statistically distinct frequencies compared to West African reference populations unaffected by the slave trade. These include stress response loci, immune regulatory markers, and variants connected to the HPA axis, the body's primary cortisol and threat response system. The field has not reached consensus on whether these frequency differences reflect selection pressure imposed by the specific conditions of enslavement, malnutrition, extreme physical labor, the documented psychological architecture of that system, or whether they reflect pre-existing variation that was amplified by the demographic bottleneck. The research is ongoing. The debate among researchers is genuine and unresolved. What I will tell you is the question being asked. Researchers at serious institutions, working with serious data, are now asking whether some of what is biologically expressed in living African American bodies is not only the record of where their ancestors came from, but the biological record of what their ancestors were forced to survive. That question may not be answered in our lifetimes, but it is scientifically askable now. And the fact that it is being asked at all is a direct product of the same genomic revolution that returned 41,799 names from a single Tuesday afternoon query. Cambridge, Massachusetts, August 4, 2023. 27 sets of remains, 9 million profiles in the database, 41,799 living matches. The system that buried those 27 people without names, without recorded origins, without documented children, that system was confident it had achieved erasure. It had not. The evidence was never destroyed. It was preserved in molecular structure, compounding in precision with every decade of scientific advancement, waiting for the question to be asked. The Catoctin Furnace study was not conducted in isolation. It was built in direct partnership with the Catoctin Furnace Historical Society and with the descendants of the enslaved community, people who had maintained the memory of this place for generations without institutional support. The 41,799 matches were not simply published in a journal. Researchers worked to reach actual descendants, to return the story to the people it belonged to. That process is ongoing, and the Catoctin methodology is now being applied at sites across Virginia, South Carolina, Louisiana, and Maryland. Each excavation, when sequenced, yields a specific genomic profile that can be matched to living descendants. The infrastructure of reconnection is being constructed in real time. There is one more thing the data has revealed, something that has received almost no public discussion. Several Catoctin individuals carry genomic profiles with deep affinity to specific West African ethnic communities that have experienced significant demographic disruption since the 18th century, through internal African migrations and colonial-era border redrawing. In some cases, the genomic profile of a person buried in Maryland in 1800 is a more complete representation of what a specific West African ethnic population looked like before that disruption than anything the modern descendants of that population currently have access to. The diaspora is preserving genomic history on Africa's behalf, not metaphorically, as literal scientific reality. Which brings us to the question the research hasn't answered yet, the one being asked in conference rooms and in the margins of published papers, but not yet stated publicly with the weight it carries. If African American ancient DNA can reconstruct pre-colonial West African population histories that no longer exist intact on the continent, what else is archived in there? What other histories are being held in the diaspora that have been lost everywhere else? The sequencing is still running. The database is still growing. The 41,799 names were found last year. The number will be different by the time you watch this. Larger. 27 people were buried under that Maryland ground. Their names were not recorded. Their origins were not recorded. The institution that put them there was certain it had erased them completely. It was wrong. By 41,799 people and still counting. The genome is the one witness that was never silenced. It has been holding this record in the molecular architecture of millions of living people for over 200 years, waiting without urgency, without judgment, for someone to finally ask what it had seen. It kept everything, and it is still talking. If this kind of forensic history is the work you want to follow, subscribe. What comes next goes somewhere most people have never been taken before.